Hi, I am Orsola Rosa-Salva from the University of Trento, and I would like to talk to you about visually naive chicks' sensitivity to motion patterns typical of agent interactions. So why do we study this in domestic chicks? Well, because we believe it's a great model to understand the origins of social predispositions, especially the role of experience in the emergence of certain social predispositions, because domestic chicks are a precocial species, which means we can hatch them in a dark incubator and we can then take them straight from the incubator to the test, and we observe their preferences the very first time they see anything in their lives. And with this method, we have found that, among other things, domestic chicks share with newborn babies some preferences to approach in the case of chicks, or to look at in the case of newborns, objects that have some cues typical of animate motion. For instance, we have a preference for objects that are self-propelled, which means objects that can start to move on their own without being pushed around by something, or for objects that have a type of motion that seems to depend on having a body structure which is typical of animals. For instance, both human newborns and chicks have this preference for point light displays that move according to a semi-rigid pattern which seems to reveal the presence of a skeleton. Or in the case of chicks, we also found a preference for motion in which the main body symmetry axis is parallel to the motion direction, and this is associated with having a bilaterally symmetric body plan, which most animals do have. So, however, the literature from human adults and human infants tells us also that there are spatiotemporal relationships in the motion of different agents. So regularities, predictable regularities in between the timing of motion of two agents, or between the position in space of two agents when they move. And by looking at those things, we can infer social interactions, we can infer goal-directed behaviors, and overall intentional behaviors. Just to cite one of the most famous examples, we can think back to the seminal studies of Heider and Zimmel, and we see these very simple geometric shapes moving on the screen, and just because of the spatiotemporal contingency, we see social interactions, goal-directed behavior, and so on and so on. So what I wanted to do here was to see whether there is a sensitivity inborn, at least in my animal model, to the spatiotemporal patterns of intentional interactions rather than social. So in the first study, we wanted simply to test whether there is a sensitivity to spatiotemporal contingencies. And in the second study, we wanted to focus on a very specific type of spatial contingency between the different agents, which is related to the orientation of the body of one kind of agents in relation to another type of agent. So, as I told you, I ran 2 studies, and each study is consisting of 6 experiments, but I will not present them all. So the methods were always the same. We took, for each of the 6 experiments, 46 naive chicks, which hatch in the darkness, in a dark incubator, in a dark room. I put them in a box. No, somebody put them in a box, actually, and brings them to the experimental apparatus, which is this long corridor with 2 openings at the 2 ends. There are 2 monitor screens that play the stimuli. One is the target stimulus, for which I predict a preference, and one is the control stimulus. So each chick is tested only once for a duration of 6 minutes, so they are visually naive at the beginning of the test, and we place them in the center of the corridor, and then there are 2 little platforms close to the stimuli, and whenever the chick wants to have a closer look to one of the stimuli, they have to climb on the platform, and then we score the first stimulus approach, which is the first platform they climb, and the percentage of time spent on the target platform over the total time spent on the 2 platforms. So very easy. So first study, we took loosely inspiration from, and very loosely inspiration from chasing stimuli to test chicks' sensitivity to spatiotemporal contingencies. So on the left, you should see the social aggregation stimulus, and on the right, the control random stimulus. Each stimulus has 4 agents, 1 big disk and 3 small disks. So the big disk can be called either the forerunner or the mother, and the smaller disks are either the chasers or the kids. So now I hope everything will become clearer. How do you start? Yep. Okay, so at the beginning, the mother is static, and the 3 kids are exploring. Look only at the social aggregation stimulus for now, it's looping. So the mother is static, and the 3 kids are exploring around in random directions. Then the mother starts to move. After a short delay, the 3 converge to a new position, and then a similar scene happens again and again. And across different scenes, the spatial way in which the agents scatter in the random phase is different, but the sequence of events and the duration of each event is always the same. In the random stimulus, we just took all of these pieces of trajectories and we scrambled them in space and time so that there was no predictable relationship, okay? So what I predicted was a preference for the social aggregation stimulus. We created the stimulus this way because we were concerned about chasing being a very predatorial stimulus. Maybe chicks will be afraid of it, who knows? This was supposed to mimic mother hen, and chicks, when the hen is there, they can explore, otherwise they can go away. Okay, what we found was a significant preference for the random stimulus. So here I'm plotting the average time, percentage of time spent close to the social aggregation, and as higher values mean more time closer to social aggregation, lower values mean more time closer to random, and as you can see, we are below chance, which is the dotted line, and the same effect was visible for the area-dependent variable. The first approach, more chicks chose first the random stimulus compared to the chicks that chose first the target. So first of all, we said, what the hell? And we replicated the experiment 1-to-1, and it came out again. No, not 1-to-1, but with a small change, but we replicated, and it came out again, the same result. So, okay, we thought this is real. So we went on, and we ran a series of controls to try to figure out why do they prefer the random stimulus. And first, we just presented the same scenes, but canceling away either the mother or the kids. And when either only the forerunner or only the chasers are present, no preference was there. So the effect seems to be driven by the relationship between the motion of the 2 agents. So we started to think that our social aggregation stimulus had a very rigid time structure, and the random stimulus not, so this might be driving the preference. So we created a random stimulus in which the direction in which the dots move was still random, but the timing and the sequence of events was, so who moves and who is static was matched. And in this case, again, the preference disappears. So to finally test this hypothesis, we basically contrasted our original random stimulus to this new temporally fixed random stimulus, and what we found was again a preference for the originally random stimulus. So they really like it more. So from this first study, we concluded that naive chicks spontaneously encode the temporal contingencies between the motion of the 2 different kinds of agents, which is already something. And we have a preference for the display where the timing of motion of one agent cannot be predicted from that of the other kind of agent. And we have 2 interpretations of this result. One is a preference for the stimulus with more behavioral variability, which could be a cue of intentional agency somehow, or an exploration of the more computationally challenging stimulus, similar to what has been found in some chasing studies with adults and older kids, that they look actually longer after the initial phase to the random. So, second set of experiments, much simpler. We decided to exploit the wolf pack effect that has been found in the human literature, and here it is. The disk is the sheep, the arrows are the wolves. They are actually moving randomly around the screen. The only thing is that the arrows are somehow magnetized in such a way that their tip points always towards the disk. So the narrow end of their main body symmetry axis points to the disk. And human adults perceive this as a very intentional, interactive, socially coordinated display, usually described as a pack of predators stalking a prey. And this seems to have some automatic attention capture, so it interferes with other unrelated behavioral tasks. So what we did was to try to adapt the task to chicks. On the left, you can see a sorry, moving? Yeah, okay. On the left, you can see a wolf pack stimulus, and on the right, we borrow again this very elegant perpendicular control stimulus in which the arrows are also magnetized, and they are also highly socially coordinated units, so the amount of regularity is the same in both displays, but the arrows of the perpendicular control just offer their side rather than the tip to the disk. And in this case, we found a preference for the Wolfpack stimulus. It was not such a strong preference as in the first set of studies. Here I'm splitting the percentage of time spent close to Wolfpack minute by minute, because we found a significant effect of time during the test. So the behavior of the chicks changed during the time, and at the end of the test, a significant preference for the Wolfpack emerged. Similar but not significant trend in the first approach. Once again, we wanted to know whether this was real, so we replicated, exactly in this case, 1-to-1, same stimuli, same everything, and the effect came out even clearer. In this case, we were more lucky. Same effect, basically, just a bit stronger. So we ran overall 6 experiments that I schematize here. The first two are these 2 red disk replicas. And then in the different experiments, we manipulated the color of the disk, because I don't have the time for the details, but we wanted to control for the visibility of the arrows when they overlap with the disk. So experiment 2 was probably a mistake. We used this very thin circle instead of a disk, and the disk is really not so visible. Experiment 3 uses a purple disk, and this shade of purple, we know chicks like it very much for imprinting as much as they do like red, so for the affiliative responses. And then we use a black disk, and this we replicated 2 times just to be sure. So these are the overall results. As you can see, basically, with the exception of experiment number 2, which is the third bar, which is the empty circle, we found a consistent, even though not very strong, preference for the Wolfpack. So we decided collectively that the best analysis for this kind of experiment was probably a meta-analysis. So here I am plotting a forest plot of the effect size with confidence intervals, and on the bottom you can see the combined effect size. So we have a small but significant overall preference for the Wolfpack stimulus. And from this set of studies, we conclude that there is an innate encoding of motion cues within in the human perception are associated to the presence of social interaction and goal-directed behavior, that there is a spontaneous encoding of both the temporal and the spatial contingencies between the motion of different agents in our naive animal model. And it's particularly interesting for me that the second study reveals an innate sensitivity to the reciprocal body orientation of our agents. And with that, I would like to thank you for being still here, the organizers for letting me talk, and all the co-authors. Most of us are from the University of Trento, but Mikolaj Hernik is actually from the Arctic University of Norway.
Thank you very much, Ursula. We have time for questions.
This is a very simple question, just why? So why do they prefer the Wolfpack stimulus? You said something about goal-directed behavior. It's not even clear what the goal is here, except that they are turning towards the target, but they are not approaching the target. But even if that's the case, why do young chicks prefer this kind of stimulus?
Well, that's a good question because I believe, well, in the natural environment, agency and animacy, they go together usually in the same kind of entities, which is the other animals. And what we believe is that there is a mechanism for the attentional prioritization of all this class of entities. Of course, normally you would have cue of self-propulsion together with all the other cues and will point in the attention. I mean, it could have been that we would not find anything, and in this case, it would mean that for the chick it's sufficient to have a detection of for instance, self-propulsion and the other cues that are described to detect the potential social partners or interesting animate creatures in its environment, and then that will be it. However, I don't...
So let me reformulate. If you find it here, the preference, why don't you find in the first study where the goal-directed behavior is much more clearer in terms of approach? It's not just turning, rotating towards a particular target, but actually approaching it, right? That would be the proper goal-directed behavior. So why do you find it in the second one and not in the first one?
So I believe that in the first one, we made a mistake when we designed the stimuli, and we created them to be really, really rigid. Like really, really repetitive. And I believe this has overridden any perception of the fact that there was too much regularity. And
so it was perceived as a mechanical...
yes, and they perceive it to be some sort of mechanical inanimate event going on and not some living things interacting.
Thank you very much. Let's thank Orsola once again.